Bismillahirrahmanirrahim. Assalamu alaikum everyone. This is Muhammad Hakim, student of MPhil first semester, Department of Microbiology, Kohart University of Science and Technology. I am going to present topic of my presentation, molecular detection of Bacillus careus and milk by polymerase chain reaction. It is been taken from research article. This is the research article from which I prepare my presentation. This research article published in International Journal of Life Sciences and Biotechnology 2021. Here is some outline of my presentation topic. We will go through introduction, material and method, result, conclusion, and last reference. Introduction. Food-borne pathogenic microorganisms cause several losses such as disease and deaths, also leading to public health problems that can reach epidemics by contaminating the food at any stage of the process from food production to consumption. It is known the raw milk and its products are one of the main causes of foodborne disease associated with Listeria monocytogenes, Staphylococcus aureus, Escherichia coli 0157H7. These two are the strain of Escherichia coli, pathogenic strain of Escherichia coli, Bacillus cereus, Salmonella species, Bacillus careus, Salmonella species, Campylobacter species, Clostridium botulinum. These all are responsible for foodborne illness. Milk is a nutrient-rich food. It is a great place in human nutrition because in milk variety of vitamins, protein, calcium, carbohydrates are present. Introduction is continued. The enzyme production of saccharophilic, lipophytic, and proteolytic bacteria and change in milk are the results of long-term storage of raw milk before pasteurization. Saccharophilic, a cool-loving bacteria, a cool-loving minimum and maximum growth on 0 centigrade to 20 centigrade. Bacillus careus bacteria cause food illness due to their enterotoxin and emetic toxin content. Non-hemolytic enterotoxin, hemolysis, and cytotoxin K are enterotoxin originated from Bacillus careus. They cause food poisoning. The emetic toxin is a peptide kerolide. Kerolide basically is a cytotoxin which should destroy mitochondria. A thermostable and acid-resistant dipeptide. Disease from Bacillus careus. Bacillus careus can be classified as a non-gastrointestinal and gastrointestinal disease. Gastrointestinal diseases can be seen into two different waves. One is diarrhea, another is emetic form. Determination of foodborne pathogens is important for accurate and standardized clinical diagnosis in the food industry and monitoring food hygiene. Why determination of foodborne pathogen is important? Because it's concerned to public health. Recently, polymerase chain reaction (PCR), ELISA, enzyme-linked immunosorbent assay, commercial kits, and tests based on different cell cultures are used to diagnose and identification of Bacillus careus toxin and foods. Next is materials and methods. There are 4 steps involved. First of all, sample collection. Another is production of bacteria and culture condition. Then DNA extraction, and then polymerase chain reaction. Sample collection. Sample collection is necessary for every experiment to proceed the process. Next is the production of bacteria and culture condition. Uh, is a positive control Bacillus careus ATCC 11778 strain was used. Then the spreading plate method was applied using selective agar. Name is MYP stand menthanol egg yolk polymyxine for bacterial cultivation of Bacillus careus. This is the selective agar for Bacillus careus. The bacterial culture was placed in incubator at 37 centigrade and left for 2 days of incubation. Bacterial colonies observed breeding was detected morphologically. Then the morphologically selected bacterial colony was taken into LB broth medium and incubated at 37 centigrade for 150 revolution per minute overnight. Next is after culturing of bacteria, next step is DNA extraction. The sample containing the bacterial colony was incubated the night before, collected by centrifugation. DNA's blood and tissue kit. These two are DNA's blood and tissue kits. These two are commercial kit for DNA for rapid application of DNA was applied according to the manufacturer instructor for bacterial colonies. DNA solution. After DNA solution, purified DNA sample were stored at -20 centigrade and the elevation buffer for residue. After DNA extraction, next step is polymerase chain reaction. Uh, Polymerase, there are three steps and PCR. One is the first one. The first one is denaturation. Another is annealing, and the last one is extension. Uh, Requirements for PCR: DNA templates, take polymerase, DNTPs, buffer solution, ions. The polymerase chain reaction PCR process was performed using Biorad C1000 touch thermal cycler. Biorad is a company name. A PCR was performed by optimizing reaction condition for each primer, and the PCR reaction were formed in a total volume of 25 microliter containing 0.04 IAC 16S rRNA or 06 HB IAHB IC HBID NHEA NHEB NHEC. These are the primers. The PCR cycle reaction are as follow: 10 minutes in first denaturation at 95 centigrade, then 94 centigrade for 60 seconds, and then 50 60 second for then 50 60 second for 60 seconds, then 50 60 centigrade for 60 seconds. The annealing temperature changing from each primer. Second step is annealing. Annealing basically done at 72 centigrade for 60 seconds. And then the third step is extension step of for 10 minutes. Then thermoscientific EC 100 XL power supply electrophoresis system was used to execute DNA sample. Biorad chemodog imaging system UV trans illuminator was used for imaging the gel where DNA and PCR were operated. PCR fragments were run for 30 minutes at 100 volts and 0.5x TBE buffer solution with 2% agarose gel electrophoresis aid with ethidium bromide and visualize under UV light. Ethidium bromide basically we aid for visualization of DNA. These are the material and method of Bacillus careus results. Table number 1, expected band size and sequence information of primer used. In table number 1, there is information about primer's name, primer sequence, and expected band size, or a band size for bacillus, carriers, and target regions. Primer's names, primer name in which we used in PCR process for target region of bacillus carriers. First, IACFF for forward, IACR for reverse, HB1A1, HB1A2, 16SF, 16SR, 45C1, 45C2, NHEBC2, NHEBC1, L1A forward, L1A reverse, L2A forward, L2A reverse. These are the primers which are used in PCR for target region of Bacillus cereus. Here is the primer sequence, expected size, and target regions. Expected band size for primer number 1, 100, 118, and for primer number 2, 874, for primer number 3, 267, for primer number 4, 186, for primer number 5, 300 base pair, and for primer number 6, 205, and for primer number 7, 411 base pairs. And there is a target region, AAC gene, HB1A, 16S RNA, NHEA, NHEB, NHEC, HBID, HB1D, sorry, and HB1C. These are the target region. Here is the table number 2 and a result portion. This table shows the number of milk sample and positive and negative result after performing PCR. 13 sample were collected. PC for positive control. In positive control sample, all primers are positive. Also, all primer are positive in sample number 1. In sample number 2, first primer are positive, others are negative. In sample number 3, also all primers are positive. In sample number 4, first primer are positive, others are negative. In sample number 5 and 6, all primers are positive. In primer number 7 and 8, first primer are positive, others are negative. In sample number 9, 10, 11, all primers are positive. In sample number 12, first primer are positive, others are negative. And sample number 13, all primers are positive. It is a visualized picture of 16S forward and 16S reverse primer after forming gel electrophoresis. Here we target IAC gene with the help of 16S F and 16S R primer. This is the another visualized picture of HB1A1, HB1A2 primer after performing gel electrophoresis. Here is the conclusion of my presentation topic. In this study, a molecular determination was made using the PCR method and primer specific to the gene region of Bacillus carriers bacteria that cause negative effect in raw and pasteurized milk samples. For this purpose, 6 primers used. It's specific to 7 toxin gene region of Bacillus carriers bacteria was performed molecular screening. Bacillus cereus bacteria were detected molecularly and 13 milk samples examining due to the study. Bacillus cereus bacteria were detected molecularly, which cause adverse effects such as food poisoning and loss of quality in milk, also cause severe disease. It is predicted that the result obtained and the method used will also help in future study for the detection of Bacillus cereus bacteria in milk and dairy products. Here is the reference of my presentation. Thank you for watching.